I'm Bérénice Batut, and today with Angie Ansar, Anna Henniger, Tobias Schindler, Paul Zeriep, Björn Gröning from either the Freiburg Galaxy team in Germany or Biolytics company in Switzerland. I present this poster, accessible and scalable pipeline for fast and easy foodborne, or not just foodborne, pathogen detection and tracking. What is the context of that work? So food contamination by pathogen affect around 600 million people per year and have socioeconomic impacts on the development at different levels. And during the investigation of a foodborne outbreak, usually a microbiological analysis of the potentially responsible food is performed to detect the responsible pathogen, identify the contamination source. Usually with traditional methods, we need to do an isolation of the targeted pathogen, which can be time consuming, not always straightforward and or not successfully. And metagenomic approaches could solve this issue by giving an overview of the genomic compositions of the sample, including what the food itself, what is the microbial community within the food, and any possible pathogens. It doesn't need any isolation of the pathogen before. It's not limited to specific genes, and it can be more accurate. If we combine metagenomic approach with Oxford nanopore sequencing, we can make the identification of pathogen quicker, easier, more accessible, and more practical. The only thing that is still missing is how to analyze such data, because it can be really complex and it lacks accessible, easy to use, and openly available pipelines. So to solve this issue, we implemented a series of fair Galaxy-based workflow that integrate state-of-the-art tools, visualization, and report to track, detect, and track pathogen from any Oxford nanopore metagenomic sample, not only food. How it looks like. So if you have something that is contaminated, you can extract the DNA directly, sequence it directly, and then run a series of bioinformatics pipeline that are made of 5 different workflows. One step is the pre-processing for checking the quality control to improve the quality of the data. Then we have a step of taxonomic profiling with Kraken to give an overview of the microbial profiling, then a gene-based pathogen identification using AMR and virulence factor genes, and that is also used then for tracking the pathogen among samples. And we have also a SNP-based pathogen identification if we expect we have an idea about the pathogen, for example, Salmonella, we do a SNP calling, and then we can have better full genomes for the different there, the consensus genome there. All these pipelines are using open source tools that are packaged using either biocontainers or Bioconda. The pipelines are built on Galaxy, so it has a user-friendly workflow environment. It's also freely available to be run on the European Galaxy server. They are annotated, shared, and deployable, and available on Workflow Hub and Dockstore, so that they can be installed on any Galaxy instance. They have been tested with more than 100 samples. I will discuss about that later, and they are documented, and especially with tutorials that go either step by step or running workflow by workflow, and that is available online on the Galaxy training website. So the results, so we tested this workflow on more than 100 samples with two different data sets. One where we spiked the food with different Salmonella enterica strains at different concentrations. And then on the first image in the middle here, we showed here the report from Kraken that see the different abundances of the different subspecies, and we clearly see two groups and where depending on the concentration there, and also what are the expected subspecies there. We also got data that has been collected in Palestine from human chicken stools and meat, and where that we know it contained either Salmonella enterica or Campylobacter jejuni, And we can see with the heat map below there, where you see the different virulence factor, that you have two groups of samples. So the samples are the different rows, and the column are are different virulence factors. We clearly see two groups of samples, one where we see the virulence factor only for Salmonella, and the other one for Campylobacter jejuni. And that are the type of outputs we can get from the workflows. So this work is supported by, has been funded by EOSC Life. And if you want to know more and have more, all the links to the different materials or the workflows, the training materials, the data analysis part that is a link there, you can follow this QR code there. And I would like to thank you there.